Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
It's really kind of wild when you think about it. Prostate cancer is the second leading cause of cancer deaths in men. And, it's especially tough when the cancer just stops responding to the treatments.
Yeah, it's a real problem. Initial therapies, they can work great for a lot of folks, but unfortunately, it often comes back, even stronger.
Like it's learned how to fight back against the drugs.
Exactly. Castration-resistant prostate cancer, it's what we call it. And it's a huge challenge, finding new ways to fight it. That's what we really need.
And that's precisely what we're diving into today.
Buckle up.
Some really interesting research out of UCSF, University of California, San Francisco. They're looking at aggressive prostate cancer from a whole new perspective. Like going right down to how cells make proteins.
The building blocks,
Exactly, the fundamentals.
And we've got two great sources for this. A super clear health news article that lays it all out.
The big picture.
And then the actual scientific paper, it's in Cancer Cell, and that one, it's a deep dive, all the nitty-gritty details.
Perfect combo.
Yeah, gives us the full story.
So before we get into the specifics of this study, let's talk about the problem they're trying to solve. What's the core issue with cancer when it comes to this protein production stuff?
Well, the news article explains it really well. You got to think of our genetic information, these things called messenger RNAs or mRNAs, right? They're like the instruction manuals for building proteins inside our cells.
Blueprints.
Exactly. And usually those blueprints, they're, carefully controlled.
Like only used when they need to be.
Right. But in cancer, certain enzymes, these things called helicases, they get out of control.
Uh-oh.
Yeah, they basically unlock tons of these mRNA instructions all at once.
So it's like the cell's just churning out proteins like crazy, nonstop.
Total chaos, like a factory on overdrive. And that's a big part of why cancer grows so fast and spreads so easily.
Makes sense. So the UCSF scientists, they honed in on one particular helicase. It had a pretty complicated name, eukaryotic initiation factor 4A.
Oh yeah, try saying that 3 times fast.
No kidding.
Yeah.
But it sounds like this one is like super important in all this protein mayhem.
It's a key player for sure.
So what did they find? What's the solution they're looking at?
Well, they were working with this drug called Zotatifin. Originally, it was developed for breast cancer.
Okay.
But get this, they discovered it does something really unique in prostate cancer. It doesn't just block this eukaryotic initiation factor 4A.
Like just shut it off.
No, it actually disables it.
Disables it.
Yeah.
So how does that work?
It transforms it, and this is straight from the news article. They call it a molecular break.
Molecular break? Okay, I'm intrigued.
They even made a picture to show it. You've got Zotatifin, that's shown in white, and it's attaching itself to the eukaryotic initiation factor, 4A enzyme, that's purple, and then there's the mRNA message that's green.
I like how they use colors. It makes it easier to visualize.
Yeah, totally. So basically, the Zotatifin sticks to the enzyme and the mRNA, and that prevents the cell from reading that mRNA.
So no protein gets made.
Exactly. The enzyme's stuck. It can't do its job.
Molecular break.
Right. Clever, huh?
I like it.
So the UCSF team, they really wanted to understand exactly how this works in prostate cancer. Can it really stop those cancer cells from growing? Could it be a real treatment, especially for the toughest one, the castration-resistant kind?
The big one.
Yeah.
So let's get into the study itself. What was their main goal?
Well, they really wanted to see how Zotatifin goes after prostate cancer, can it actually stop those cancer cells from growing and stuff? And, they especially wanted to see if it could be a treatment for castration-resistant prostate cancer.
The one that doesn't respond to the usual treatments.
Right. Tough one.
Okay. And we've talked about this eukaryotic initiation factor 4A enzyme a bit, but why were they so focused on that specifically?
Well, the scientific paper goes into a lot more detail about that. It explains that this eukaryotic initiation factor, 4A, it's an enzyme, a helicase enzyme, and it's really important for unwinding these, tangled up parts of the mRNA molecules.
So, it smooths out the instructions?
Yeah, like ironing out wrinkles so the cell can actually read the mRNA and start building the protein.
Okay.
The thing is, cancer cells, especially prostate cancer cells, they get kind of addicted to this eukaryotic initiation factor 4A.
Like they needed to survive.
Right. They need it to make all the proteins that help the cancer grow.
So the more of this enzyme, the worse the cancer.
That's what it looks like. They actually looked at tumor samples from 500 patients at UCSF, all with prostate cancer. And as the cancer got worse, the levels of this enzyme, it just kept going up.
Oh, wow.
Yeah, so it seems like a pretty good target for new treatments.
Makes sense. Now, we know Zotatifin kind of gums up the works with this enzyme, but how does it actually do that? We know it binds, but then what?
So the scientific paper, it clarifies that Zotatifin, it basically grabs onto both the enzyme, the eukaryotic initiation factor 4A, and the mRNA at the same time.
Like a double whammy.
Yeah, it jams up the whole system.
Yeah.
The mRNA can't get translated into a protein, and the enzyme, it just sits there, can't do its job, stuck, like that molecular break we were talking about.
Pretty cool. But they didn't just do this in a dish in the lab, right? They did some experiments with mice too, didn't they?
Oh yeah, they use these things called patient-derived xenografts.
Xenografts, what are those?
So it's a really cool and important way to test cancer drugs.
Okay.
They take actual pieces of prostate tumors directly from patients, and they implant them into mice so they can see how the drug works on real human tumor tissue.
So it's like as close as you can get to testing it in a human without actually, testing it in a human.
Exactly. Gold standard for preclinical research.
Okay. So what happened when they gave these mice with the human tumors Zotatifin?
Well, the news article, it highlights this part really well. The tumors that had been growing in the patients, they actually shrank in the mice that were treated with Zotatifin.
Really? That's impressive.
Yeah, it was a good sign that the drug might actually work.
So were all the prostate cancers in the mice the same?
No, they actually tested it against a bunch of different models. They had primary and metastatic hormone sensitive, so that means they still respond to the hormone-blocking treatments.
Okay.
And then they had a castration-resistant model called LTL-484, the really aggressive kind.
The tough one.
Yeah.
And it worked on all of them.
Yeah, pretty much. Every model they tested, the Zotatifin, it made the tumors grow slower, and the mice lived longer too. Plus, they didn't lose a bunch of weight, which means the drug probably wasn't causing a ton of side effects.
That's good.
At least in mice, early days.
Yeah. And I think they also used some regular lab-grown cancer cells in mice too, right?
Yeah, they did. They call those cell line xenografts, and they injected those into the mice. So they used these lines called 22RV1, C42B, and VCAP, all castration-resistant types. And then they used another one, LNCAP, but they injected that directly into the prostate of the mice.
Oh, wow. So really specific placement?
Yeah, orthotopic injection, they call it.
Fancy.
Yeah, well, science.
So, and did the Zotatifin, did it work there too?
It did, every time. Reduced the tumor burden, they call it, and the mice lived longer. More evidence that this could be a real treatment.
Exciting. So they also looked at some specific proteins that are known to drive prostate cancer. What did they find out about those?
Right. So they focused on two big ones. First, the androgen receptor, which is a major fuel for prostate cancer growth. A lot of the current treatments, they try to block this receptor. Yeah. But with Zotatifin, it actually lowered the amount of the androgen receptor protein in the cells.
So less fuel for the cancer.
Exactly. And get this, it even seemed to stop the cells from making the androgen receptor in the first place.
Like cutting it off at the source.
Right. Messing with the mRNA, just like we talked about. And the scientific paper, it explains how this happens.
Fill us in.
So when the eukaryotic initiation factor 4A with the Zotatifin attached, it tries to unwind the mRNA for the androgen receptor, but it gets blocked, it can't do it.
So the mRNA can't get read.
And no androgen receptor protein gets made.
Wow. Now, there's that tricky version of the androgen receptor, the ARV7, right?
Mm-hmm.
The one that doesn't respond to the usual hormone treatments?
Yeah, that one's a real troublemaker. It's always on, even when we're trying to shut it down. And that's a big reason why some prostate cancers become resistant.
Super frustrating.
Totally. But good news, the Zotatifin, it actually reduced the amount of this ARV7 protein too. And for both the regular androgen receptor and ARV7, it lowered the protein levels without changing the mRNA levels.
Interesting.
Yeah, so it's working directly on that translation process, right? Not at the gene level.
So it's a more precise intervention. And did this reduction in the androgen receptor protein, did it affect anything else? Like downstream?
It did. It did. They looked at PSA, prostate-specific antigen.
The marker for prostate cancer.
Right, and that's controlled by the androgen receptor.
Okay.
So they found that Zotatifin treatment, it actually decreased the PSA levels too, which means overall, the androgen receptor wasn't working as much. And they did this bigger analysis too, looking at a whole bunch of genes that the androgen receptor controls, and they found that its activity, it just went down across the board.
So it wasn't just about the amount of the receptor protein itself, it was about actually reducing its influence.
Yep, taking away its power.
Cool. Okay, what about that other protein, the hypoxia-inducible factor 1 alpha? What's that all about?
Right, so that one, HIF-1A for short,
HIF-1A, got it,
it helps tumors survive in low oxygen environments.
which are common in tumors.
Right, it's called a hypoxia,
Yeah.
and this HIF-1A, it's also linked to resistance to radiation therapy.
Uh-oh, another troublemaker.
Yeah, kinda, but just like they saw with the androgen receptor, Zotatifin, it also decreased the levels of this HIF-1A protein in their models.
So it was like a 2-for-1.
Yeah, pretty much.
And was this happening because of the mRNA levels again?
That's the interesting part. No, not really. The mRNA levels, they either didn't change much or even went up a little bit with Zotatifin.
So it was specifically targeting the translation process again.
Exactly. And they saw this effect on HIF-1A in both the lab-grown cell models and the ones with the patient tumor pieces, especially when there was low oxygen.
When HIF-1A is most active.
Right.
So Zotatifin is going after these two big drivers of prostate cancer by messing with protein production. Sounds like a pretty big deal.
Huge implications. The researchers, they actually use this term translatome therapy.
Translatome therapy.
It's basically treatments that specifically target how cancer-causing proteins get made.
Okay.
And they say that Zotatifin, it's a perfect example of this, like a whole new category of drugs.
So could we combine Zotatifin with other treatments too, like the stuff we already use?
Oh, absolutely. They tested a few combinations in their preclinical models.
How nice.
Yeah, so first they tried it with anti-androgen drugs, like enzalutamide and darolutamide. These drugs, they block the androgen receptor's activity.
Okay.
The idea was if you use Zotatifin to lower the amount of androgen receptor, and then you hit it with these other drugs to block whatever's left.
Double whammy again.
Right. So in the models, they found that the combo, it was way more effective than either drug alone, shrunk the tumors more, and the mice lived longer.
Sounds promising. What other combos did they try?
Well, they also looked at combining Zotatifin with an AKT inhibitor called capivasertib.
AKT inhibitor, okay. What's the logic there?
So Zotatifin, it also affects this pathway called PI3K-AKT, which is involved in cell growth and survival. And this pathway, it's often like super active in prostate cancer. Capivasertib, it blocks AKT, which is a key protein in this pathway. So they thought maybe if you use Zotatifin to lower the amount of another protein in the pathway called PIK3CA, and then you use capivasertib to block the activity, you might get a better response.
Makes sense, like attacking it from two angles.
Right. And it worked, in their models at least. Especially in the ones with a lot of PIK3CA. The combo, it really held back the tumor growth.
Wow, so they also tried it with radiation therapy too, right? That's pretty standard for prostate cancer.
They did.
Why that combo?
Well, you know how we were talking about low oxygen areas in tumors?
Yeah.
They can make the cancer cells resistant to radiation.
Right.
So since Zotatifin lowers HIF-1A, that protein that helps cells survive with low oxygen, they figured maybe Zotatifin could make the cells more sensitive to radiation.
Make the radiation work better.
Exactly. It worked. In the lab, Zotatifin made the cells more vulnerable to radiation, especially with low oxygen. And in the mice, the combo of Zotatifin and radiation, it really slowed down the tumors and the mice lived longer.
So maybe a way to deal with that pesky resistance.
Could be, could be.
Really interesting stuff. And, beyond the treatment itself, could this research help us figure out which patients might benefit most from Zotatifin? Like, is there a way to personalize it?
Yeah, that's the whole biomarker idea. And the researchers, they actually found that high levels of that eukaryotic initiation factor, 4-sapatoprotein in tumor samples, it was linked to a higher risk of the cancer spreading.
Metastasis.
Right, and a higher chance of dying from the cancer after surgery. So maybe if someone's cancer comes back checking their tumor for high levels of this enzyme, it could help doctors decide if Zotatifin might be a good option.
Personalized medicine.
Right.
Of course, it's early days. What are some of the limitations the researchers themselves brought up?
Well, first off, they were very clear that the results, the mouse studies and the lab-grown cells, it's all very exciting, but it needs to be tested in actual humans with prostate cancer.
Clinical trials.
Yep. And Zotatifin, it is in trials for breast cancer and some other solid tumors, but nothing specifically for prostate cancer yet.
So we got to see how it does in people.
Absolutely. And they also pointed out that their study, it mainly focused on the androgen receptor and HIF1A,
Right.
but they found some other potential targets too, like SLC2A1, which helps with glucose transport, and CDK1 and CDK4, which are involved in the cell cycle.
So the Zotatifin might be doing even more than we realize.
Could be. Could be. They definitely say we need to do more research on those other targets.
Makes sense. What about the specifics of how Zotatifin messes with the mRNA? Like the details of that?
Yeah, so they showed that Zotatifin binding, it leads to changes in the structure of the mRNAs for the androgen receptor and HIF1A, especially in these regions called the 5' untranslated regions. But they admit that the exact way this binding causes those changes and how that stops the translation process, it still needs more study.
So it's like, we know it's messing with the folding, but we don't know exactly how it's doing it.
Right. And there were some sequences in the androgen receptor mRNA that they thought were important, but they turned out to not really matter that much.
Okay.
So there's definitely more to figure out.
Always more to learn.
Oh.
But the researchers, they seemed pretty optimistic about all this, right?
Oh, yeah, definitely.
What were some of the key things they said?
Well, Dr. Devi Ruggiero, he's one of the senior authors, he pointed out that prostate cancer really exploits this eukaryotic initiation factor 4A, to get around the normal controls on protein production.
Speaking.
Yeah. And he said that having a drug like Zotatifin, something that can specifically target this enzyme, it could be a real game changer for prostate cancer treatment.
Strong words.
Big hopes.
What about Dr. Digu Kuzuglu Ozturk?
She was one of the lead authors. She called Zotatifin a bona fide translatome therapy, something that directly blocks the production of those bad proteins.
Right.
And she said that this whole new class of drugs, it really opens up the possibilities for treating cancer.
More tools in the toolbox.
Exactly.
And Dr. Howden Gwani, who was another lead researcher, what did he think?
He was really hopeful that Zotatifin could make treatment more durable, so it lasts longer and the cancer doesn't come back as quickly.
Fingers crossed.
Yeah. And he said that this research, it gives hope to a lot of patients who are fighting this disease.
Much needed hope. So overall, pretty positive outlook.
Yeah, definitely. They even specifically said they hope this will lead to clinical trials for Zotatifin in prostate cancer.
So fingers crossed for that. This has been a fascinating deep dive into a whole new way of looking at prostate cancer treatment. It seems like targeting the protein production process itself, it could be a real breakthrough. So as we wrap up today, what's the big takeaway you'd like our listeners to ponder?
I think it's this. When we really get down to the nuts and bolts of how cancer works, like the really fundamental stuff, it can open up completely new ways to fight it. We might not have even thought of these strategies before. And it makes you wonder, what other diseases could we tackle this way, beyond cancer? Maybe by targeting protein production, we could find new treatments for all sorts of things.
Wow, that's a great point. So much potential. Now, for our listeners who want to learn more, where can they go?
Well, the full research paper, it's in Cancer Cell. You can find it with the DOI 10.10116J.C-cell.2.225/02.027. We'll put that in the show notes, too. And, of course, there's that great health news article from UCSF that gives a nice overview.
Perfect. Well, thanks for joining us for this deep dive. We'll keep our eyes peeled for any updates on Zotatifin and those clinical trials for prostate cancer. So all our listeners, stay curious.
Stay informed.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.